Hemostasis. Textbook. Guyton and Hall. Textbook of Medical Physiology. 11th edition. Pages 457 to 468. Chapter 36. Hemostasis and blood coagulation. Part 3. Prevention of blood clotting in the normal vascular system. Intravascular anticoagulants. Endothelial surface factors. Probably the most important factors for preventing clotting in the normal vascular system are 1. The smoothness of the endothelial cell surface, which prevents contact activation of the intrinsic clotting system. 2. A layer of glycocalyx on the endothelium. Glycocalyx is a mucopolysaccharide adsorbed to the surfaces of the endothelial cells, which repels clotting factors and platelets, thereby preventing activation of clotting. And 3. A protein bound with the endothelial membrane, thrombomodulin, which binds thrombin. Not only does the binding of thrombin with thrombomodulin slow the clotting process by removing thrombin, but the thrombomodulin-thrombin complex also activates a plasma protein, protein C, that acts as an anticoagulant by inactivating activated factors 5 and 8. Endothelial surface factors. When the endothelial wall is damaged, its smoothness and its glycocalyx thrombomodulin layer are lost, which activates both factor 12 and the platelets, thus setting off the intrinsic pathway of clotting. If factor 12 and platelets come in contact with the subendothelial collagen, the activation is even more powerful. Antithrombin action of fibrin and antithrombin 3. Among the most important anticoagulants in the blood itself are those that remove thrombin from the blood. The most powerful of these are 1. The brin-burs that themselves are formed during the process of clotting, and 2. An alpha-globulin called antithrombin 3 or antithrombin heparin cofactor. While a clot is forming, about 85 to 90% of the thrombin formed from the prothrombin becomes adsorbed to the brin-burs as they develop. This helps prevent the spread of thrombin into the remaining blood and, therefore, prevents excessive spread of the clot. The thrombin that does not adsorb to the brin-burs soon combines with antithrombin 3, which further blocks the effect of the thrombin on the brinogen and then also inactivates the thrombin itself during the next 12 to 20 minutes. Heparin. Heparin is another powerful anticoagulant, but its concentration in the blood is normally low, so that only under special physiologic conditions does it have significant anticoagulant effects. However, heparin is used widely as a pharmacological agent in medical practice in much higher concentrations to prevent intravascular clotting. Heparin. The heparin molecule is a highly negatively charged conjugated polysaccharide. By itself, it has little or no anticoagulant properties, but when it combines with antithrombin 3, the effectiveness of antithrombin 3 for removing thrombin increases by 100-fold to 1,000-fold, and thus it acts as an anticoagulant. Therefore, in the presence of excess heparin, removal of free thrombin from the circulating blood by antithrombin 3 is almost instantaneous. The complex of heparin and antithrombin 3 removes several other activated coagulation factors in addition to thrombin, further enhancing the effectiveness of anticoagulation. The others include activated factors 12, 11, 10, and 9. Heparin is produced by many different cells of the body, but especially large quantities are formed by the basophilic mast cells located in the pericapillary connective tissue throughout the body. These cells continually secrete small quantities of heparin that diffuse into the circulatory system. The basophil cells of the blood, which are functionally almost identical to the mast cells, release small quantities of heparin into the plasma. Mast cells are abundant in tissue surrounding the capillaries of the lungs and to a lesser extent capillaries of the liver. It is easy to understand why large quantities of heparin might be needed in these areas because the capillaries of the lungs and liver receive many embolic clots formed in slowly owing venous blood. Sufficient formation of heparin prevents further growth of the clots. Lysis of blood clots plasmin. Lysis of blood clots plasmin. The plasma proteins contain a euglobulin called plasminogen, or probrinolysin, that, when activated, becomes a substance called plasmin, or brinolysin. Plasmin is a proteolytic enzyme that resembles trypsin, the most important proteolytic digestive enzyme of pancreatic secretion. Plasmin digests brin-burs and some other protein coagulants such as brinogen, factor 5, factor 8, prothrombin, and factor 12. Therefore, whenever plasmin is formed, it can cause lysis of a clot by destroying many of the clotting factors, thereby sometimes even causing hypocoagulability of the blood. Activation of plasminogen to form plasmin, then lysis of clots. When a clot is formed, a large amount of plasminogen is trapped in the clot along with other plasma proteins. This will not become plasmin or cause lysis of the clot until it is activated. The injured tissues and vascular endothelium very slowly release a powerful activator called tissue plasminogen activator, TPA, that a few days later, after the clot has stopped the bleeding, eventually converts plasminogen to plasmin, which in turn removes the remaining unnecessary blood clot. In fact, many small blood vessels in which blood has been blocked by clots are reopened by this mechanism. Thus, an especially important function of the plasmin system is to remove minute clots from millions of tiny peripheral vessels that eventually would become occluded were there no way to clear them. Do you have any question?